You think you know your brain. You've lived inside it your entire life, treating it like a familiar home. Every thought you've ever had, every memory, every fear, every quiet moment of unexpected joy, all of it happened in there. It feels intimate. It feels like home. It feels, most of the time, like you. But here is the thing no one tells you. The moment you zoom past the elegant branching arms of the neuron and cross the quantum threshold, the brain stops looking like a biological organ. It starts looking like the inside of a star, something ancient, operating on ancient software written before the dinosaurs, software that has never been updated. Tonight, we go all the way in, past the neuron, past the quantum threshold, to the bottom of the abyss, where the tissue you live in is stranger than the cosmos itself. Welcome to Sleep Entangled, where science and serenity merge into contemplative voyages for the quietly curious mind. If you find wonder and depth in these explorations, remember to subscribe and let the questions guide your nights. Now settle, breathe, and let's go deeper than we've ever gone before. Chapter 1. The deep architecture, the ancient stranger within here is something worth sitting with before anything else. Right now, as you lie here, as your breathing begins to find its own quiet rhythm and the events of the day recede like a tide, there is something inside your skull that was already ancient before the first human being ever walked the earth, before language, before fire, before the first dream anyone ever had, before the first question anyone ever asked about what they were, or why they were here, or where the sky ended. It is your brainstem, and it is not entirely yours. Let's begin there, at the absolute bottom of the brain, not with the cerebral cortex, that magnificent wrinkled crown we associate with everything we consider distinctly human. Language, mathematics, art, self-reflection, the capacity to lie awake in the dark and wonder about quantum mechanics. Let's start with what came first. The brainstem is one of the oldest structures in the entire vertebrate nervous system. It sits at the base of the brain, continuous with the spinal cord, and it manages the machinery of being alive with a competence and a quiet authority that should honestly make us more humble than it does. Right now, as you listen to this, your brainstem is regulating your heart rate, modulating your breathing, adjusting the tension in your blood vessels, controlling your swallowing reflex, and coordinating the movement of your eyes as they rest beneath their lids. It is making, by some estimates, more than 100,000 micro-adjustments per hour. You were not consulted on a single one. The neuroscientist Paul MacLean, who spent much of his career at the National Institute of Mental Health, developed what he called the triune brain model, a framework proposing that the human brain is, in evolutionary terms, 3 brains layered on top of each other across hundreds of millions of years. The innermost layer, which he called the reptilian complex, comprises the brainstem and the cerebellum. It is in its architecture and its function the brain of a very old animal, a fish, roughly. The structures here are not metaphorically similar to those found in reptiles and amphibians. They're virtually identical to them. The lamprey, one of the most ancient jawless vertebrates alive today, has a brainstem that would be anatomically recognizable in a human neurosurgery textbook. Modern neuroscience has refined and complicated MacLean's clean tripartite picture. The boundaries between his 3 layers turn out to be far more porous and interconnected than he imagined, but the core observation stands. Part of you is operating on software that was written before the dinosaurs, and it has not been significantly updated. There is something quietly disorienting about this when you let it settle. The part of your brain that manages your survival, that pulls your hand from a hot surface before you consciously register pain, That floods your bloodstream with adrenaline when something moves suddenly at the edge of your vision, that keeps your diaphragm contracting even when you are completely unconscious. That part is not human in any meaningful sense. It predates humanity by a margin so large the number ceases to carry weight. Now move upwards slightly past the brainstem and you find a structure that deserves far more attention than it typically receives in popular accounts of neuroscience. The thalamus. Sitting at the centre of the brain, roughly the size and shape of a walnut, the thalamus is the great relay station of conscious experience. Almost every sensory signal arriving from the outside world, vision from the optic nerve, sound from the auditory pathways, touch and pain from the spinal cord, even taste and smell by indirect routes, passes through the thalamus before it reaches the cortex. Every piece of information you believe you are perceiving right now, every aspect of this moment as you experience it, has been filtered, prioritized, and passed through the judgment of this small oval structure that you have never once consciously thought about. The neuroscientist Gerald Edelman, whose work on neural Darwinism transformed our understanding of how the brain organizes itself, described the thalamus as a core participant in what he called the dynamic core of consciousness. Not a passive relay, an active participant in the construction of awareness. The brain, in Edelman's framework, is not a hierarchy with consciousness at the top, receiving processed data from structures below. It is a constant, rapid, reciprocal conversation between the thalamus and the cortex, a dialogue operating at frequencies between 40 and 80 cycles per second that, for reasons we still do not fully understand, somehow gives rise to the unified experience of being a self in a world. Antonio Damasio, one of the most important neurologists working today, has documented cases that make the thalamus's role viscerally clear. Patients with significant thalamic lesions can exist in a state where they are technically awake, eyes open, reflexes intact, physiologically alive, but entirely absent, not asleep, not dreaming, not in any state the ordinary vocabulary of consciousness was designed to accommodate. The technical term is thalamic coma, and it represents something that should disturb our confidence in locating consciousness anywhere simple. If you remove the thalamus from the equation, the lights go out. Not because the cortex is damaged, because the conversation that generates experience has lost its central partner. And then there is the reticular activating system, a diffuse distributed network of neurons threading through the brain stem like roots through old soil. The reticular activating system is, in the most literal sense, the biological switch for wakefulness. When it fires, you are conscious. When it quiets, as it is doing right now, as the day releases its grip and the night takes its place, you begin the long, gradual descent towards sleep. You can feel that descent happening as you listen to this. The ancient brain is beginning its evening work. The gatekeeper is lowering its guard. The reticular formation is dialing back its output, and the world is growing softer at its edges. Let this be the thought that carries you into what comes next. You are not a single self in a single brain. You are a collaboration, a negotiation between structures of wildly different ages, different evolutionary origins, different operating principles. The part of you listening to this voice right now is among the most recent arrivals in that collaboration, but it is sitting on a foundation that has been doing its extraordinary work for longer than the imagination can comfortably reach. So as you rest here, held by that ancient architecture, let the question float gently. If so much of what keeps you alive is happening without your knowledge, without your input, without your awareness, what else might be? We're only beginning to find out. Chapter 2. The Forest of Shadows, the Glial Revolution. For most of the history of neuroscience, the neuron was everything. Neurons were the stars of the story. They fired, they connected, they formed networks of staggering complexity. They transmitted the electrochemical signals that became thought and memory and perception, and everything we consider the contents of a mind. The neuron was where the action was, and everything else in the brain, the vast, dense, chemically busy cellular population that surrounded and supported the neurons, was understood to be exactly that: support, infrastructure, scaffolding. The word scientists gave these other cells comes from the Greek for glue, glia, the brain's glue. Important in the way that concrete is important to a building, structurally necessary but not the point. For roughly a century that is what the science said, and that is largely what the textbooks taught. That picture has not been refined or updated. It has collapsed. Starting in the 1990s and accelerating with real urgency into the 2000s and beyond, a sequence of discoveries began dismantling the neuron-centric model of brain function so thoroughly that some of the most respected names in neuroscience now speak not of revision, but of a genuine paradigm shift. A revolution in how we understand what thinking is and where it happens. Let's start with the numbers, because the numbers alone should have triggered this reckoning decades earlier. Glial cells do not outnumber neurons by a small margin. Depending on the brain region and the methodology used to count them, estimates range from rough parity to a ratio of 10 glial cells for every single neuron. The human brain contains approximately 85 billion neurons. It may contain as many as 85 to 170 billion glial cells. We spent a century building the entire theoretical framework of neuroscience around what may be, in cellular terms, the minority population. But the numbers are not the revelation. The revelation is what glial cells are doing. There are several types. The most numerous are astrocytes, named for their beautiful star-like morphology, with long branching processes radiating outward from a central body, like the arms of an elegant biological snowflake. Astrocytes are everywhere in brain tissue. They wrap themselves around blood vessels, maintaining the blood-brain barrier that controls what enters brain tissue from the bloodstream. They wrap themselves around synapses, every single synapse, with such consistency that some researchers now call the synapse. Not a structure between two neurons, but a tripartite synapse, the axon terminal of the sending neuron, the dendrite of the receiving neuron, and the astrocyte process that ensheathed them both. For a long time, the astrocyte was thought to be holding the synapse in place. Structural support, once again. Glue. In 1994, the neuroscientist Anne Cornell Bell, working at Yale University, published a discovery that should have changed the field overnight. She showed that astrocytes, when chemically stimulated, do not simply respond passively. They produce waves of calcium ion signaling, a kind of slow, spreading electrochemical pulse that propagates through networks of connected astrocytes at their own speed, on their own schedule, entirely independent of neuronal activity. The glia were communicating, not as a byproduct, not incidentally, communicating with dynamics that looked, biochemically, remarkably like information processing. The response from the broader neuroscience community was initially skepticism, then replication, then, gradually, a dawning recognition that something fundamental had been missed. Philip Hayden at Tufts University took the next significant step. His research demonstrated that astrocytes do not merely overhear the conversations between neurons, they actively participate in them. Astrocytes release their own signaling molecules, a class now called gliotransmitters, directly into the synaptic cleft. They influence the probability that a neuron will fire. They modulate the strength of synaptic connections. They appear to be capable of potentiating or suppressing neural activity across entire local networks in ways that are coordinated, not random. The astrocyte is not watching the performance, it has lines. Then came perhaps the most practically significant discovery in this entire revolution. Micke Nedergaard, a Danish neuroscientist working at the University of Rochester, spent years studying what happens to the brain during sleep. In 2013, her team published results that were nothing short of extraordinary. During sleep, specifically during the deep slow-wave stages that dominate the first half of the night, the cells of the brain, driven by astrocytic activity, physically change shape. They shrink. The spaces between cells expand, and cerebrospinal fluid, driven by this expansion in a pulsing rhythmic flow, moves through the brain tissue in a way that cannot happen during wakefulness, flushing out metabolic waste products that accumulate during the day's activity. Nedergaard called this the glymphatic system, the brain's lymphatic system running on glial infrastructure, and among the waste products it clears is amyloid beta, the protein that accumulates in the brains of Alzheimer's patients. The pathology of the most feared neurodegenerative disease of our time is in part a failure of the brain's nighttime cleaning system. This is what your brain is doing tonight as you listen to this voice and the day releases its hold on your muscles. Your astrocytes are at work. The cerebrospinal fluid is beginning its quiet pulse. The forest is cleaning itself. I want to stop here and speak directly for a moment. As a scientist and as someone who has spent years working at the intersection of biomedical science and traditional Chinese medicine, the glial revolution has implications that reach far beyond the structural understanding of the brain. It has direct bearing on something most of us quietly care about. The question of whether real change, deep change, the kind that rewires behavior and not merely suppresses it, is actually possible, and how it happens. In my clinical work, and in the research I've engaged with from both Western neuroscience and the Heidelberg model of traditional Chinese medicine, the same principle keeps emerging. Genuine transformation at the level of habit, at the level of chronic stress responses, at the level of deeply encoded behavioral patterns. This does not happen through conscious effort alone. You cannot think your way into a new nervous system. The thinking layer is too recent, too thin, too easily overridden by the deeper systems running beneath it. What glial neuroscience is showing us is that the brain's real plasticity, its real capacity for reorganization, operates in exactly these deeper, slower, less visible systems. The glymphatic clearing that happens during sleep is not simply waste removal. It is, in a very real neurochemical sense, the brain releasing the residue of the day's patterns and preparing the substrate for new ones. The calcium waves spreading through astrocyte networks during rest are not background noise. They are the brain doing something we are only beginning to have instruments sensitive enough to measure. This is why sleep is not passive. It is not the absence of brain activity. It is a different mode of brain activity, one that the waking state actually prevents. The glia need the waking brain to quiet down before they can do their most important work. So here is what I would offer you as you lie here, as your breathing deepens and the glial networks of your brain begin the work that only the night allows. The changes you want, the patterns you want to release, the new architecture you want your nervous system to grow toward. They are happening right now, in the part of the brain that never makes it into conversations about self-improvement, or willpower or discipline. They are happening in the forest. The forest is not still. The forest is not sleeping. It is right now exactly as alive as it has ever been, doing exactly the work your waking mind never knew to ask for. Imagine it, if you will, as you drift deeper. Not the sunlit forest of a summer afternoon, but a deep sea forest, bioluminescent, alive with slow pulses of light moving through networks of extraordinary complexity. Calcium waves spreading like aurora through the dark matter of the mind. A forest that processes, that clears, that rewires, that thinks, not in the language of ideas, but in the older language of chemistry and charge and time. You are resting in that forest right now, and it is taking care of you. Chapter 3. The Quantum Microtubules. Consciousness at the smallest possible scale. We need to go smaller, past the neuron, past the synapse, past the cell membrane itself, into the interior of the neuron, into a structure so small, so architecturally elegant, and so scientifically contentious that it has divided neuroscience and theoretical physics for 3 decades. A structure that, if a handful of serious and rigorous scientists are correct, may be where consciousness itself is actually born. We're going to talk about microtubules. Every cell in your body contains microtubules, hollow cylindrical protein structures made from a molecule called tubulin, assembled into a tube approximately 25 nanometers in diameter. To give you a sense of scale, a single human hair is roughly 80,000 nanometers wide. You could fit more than 3,000 microtubules side by side across a single hair and still have room. They are, by any ordinary measure, incomprehensibly small. In most of the body's cells, microtubules serve primarily as the cytoskeleton, the internal scaffolding that gives cells their shape and provides the structural rails along which the cell's machinery moves during division. In neurons, they do all of this too. But in neurons, because of the neuron's extraordinary geometry, those long branching axons that can extend, in the case of motor neurons, for more than a meter from the spinal cord to the tip of your foot, the microtubules also serve as transport highways, moving proteins, organelles, and signaling molecules over distances that, at the cellular scale, are essentially continental. But none of that is why we're here. In 1994, the Oxford mathematician and physicist Sir Roger Penrose and the American anesthesiologist and consciousness researcher Stuart Hameroff at the University of Arizona published a theory that remains one of the most provocative, most discussed, and most disputed proposals in the entire history of consciousness science. They called it orchestrated objective reduction, OR. The theory begins with a problem. The problem is you. Classical neuroscience, the neuroscience of neurons firing and networks activating and information flowing through cortical circuits, is extraordinarily good at describing the neural correlates of conscious experience. Show a person a red apple, and you can trace with increasing precision the cascade of neural activity from retinal photoreceptors through the thalamus, through the visual cortex, into the higher association areas that integrate color, shape, and memory. The technology for tracking this cascade improves every year. What classical neuroscience cannot do, what no existing theory of neural computation can do, is explain why any of that activity produces the experience of seeing red. Not the processing of red, the feeling of it. The felt quality of redness, which philosophers call qualia, is completely absent from every mechanistic account of what neurons do. The mechanism describes the processing. The mechanism does not and cannot on its own terms describe the experience. Penrose believed this gap was not a gap in our current knowledge that better neuroscience would eventually fill. He believed it pointed to something fundamental, something about the nature of mind that physics itself did not yet have the right conceptual tools to address. Specifically, he argued that human mathematical understanding, the kind of direct, non-algorithmic insight that allows a mathematician to perceive the truth of a statement that cannot be formally proven from within the system, could not, even in principle, be replicated by any classical computational process. The mind, Penrose argued, must be doing something that classical computation cannot do, and the only framework in physics that permits genuinely non-algorithmic, non-deterministic outcomes is quantum mechanics. Penrose and Hameroff proposed that microtubules are the site where quantum mechanical processes relevant to consciousness occur. The tubulin proteins that make up the microtubule lattice, they argued, exist in quantum superposition, occupying multiple conformational states simultaneously, in the way that quantum particles exist in superposition of multiple states before measurement. These superpositions are orchestrated by the surrounding biological environment, by the neuron's electrical activity, by the regulatory proteins that attach to the microtubule surface, and periodically, these superpositions undergo what Penrose calls objective reduction, a collapse from quantum possibility into a single classical state, driven not by any observer, but by a threshold of space-time curvature at the quantum level. That collapse, Penrose and Hameroff proposed, is a moment of conscious experience. Each instant of awareness, each discrete moment in the stream of consciousness, is a quantum collapse event occurring inside the microtubule lattice of your neurons. Consciousness is not the product of classical neural computation. It is a quantum phenomenon, rooted in the same layer of physical reality that governs particle behavior, quantum entanglement, and all the extraordinary physics we have explored together in previous episodes. The scientific response to Orch was, to put it charitably, mixed. The central objection was thermal decoherence. The brain is warm, wet, and chemically turbulent. And quantum coherence, the fragile quantum states required for the theory to work, were thought to be destroyed by thermal noise in timescales far too short for any computational role. Femtoseconds, 1/4,000,000th of a second. The brain, critics argued, is simply not a quantum computer. Then, in 2007, something changed the landscape of the entire debate. A team at UC Berkeley, led by the physical chemist Graham Fleming, published evidence of quantum coherence playing a functional role in photosynthesis, the biological process by which plants capture energy from light. The molecules involved were maintaining quantum coherent states at room temperature in a biological environment for timescales relevant to energy transfer. This should not, on the prevailing assumptions, have been possible. The theoretical framework that ruled out quantum biology in warm systems was demonstrably wrong. This discovery did not prove Orch. Let us be absolutely clear about that. But it demonstrated beyond any further reasonable dismissal that quantum mechanics can operate in biological systems under physiological conditions. The door was open. Since then, evidence for quantum effects in biology has accumulated across multiple systems. In the magnetic navigation of migratory birds, which appears to rely on quantum entanglement in cryptochrome proteins in the eye. In enzyme catalysis, where quantum tunneling dramatically accelerates reaction rates in ways classical chemistry cannot account for. In the mechanism of olfaction, where quantum vibrational sensing may explain how the nose distinguishes between molecules with identical shapes but different isotopic compositions. Anirban Bandyopadhyay at the National Institute for Material Science in Japan has spent years conducting direct experiments on isolated microtubules, measuring their electromagnetic resonance properties and finding oscillation frequencies that are consistent with, though not yet proof of, quantum coherent behavior. His work remains contested, ongoing, and genuinely important. Where does this leave us? in the only honest position available to a scientist confronting evidence at this frontier: uncertainty. Orch is not proven. It may be wrong. The quantum effects in microtubules may prove to be real, but irrelevant to consciousness. The theory may require substantial revision before it accounts for all the data, but it's a serious proposal by serious scientists, and the question it is trying to answer, why does neural activity produce experience at all, remains completely unanswered by every competing framework. So as you rest here, in the quiet dark, consider this. Inside the neurons that are right now processing the sound of this voice, inside the microtubule lattice threading through those neurons, something may be happening at a scale so small it touches the boundary between matter and possibility. Whether or not it is the physical seat of your awareness, it is real, it is extraordinary, and it is happening inside you, right now, without any effort from you at all. You are, at your smallest accessible scale, a quantum structure navigating the boundary between the determined and the possible. That is not a metaphor. It is a scientific hypothesis worth taking seriously. And tonight, as the day's noise recedes, it might be the most accurate thing anyone has ever said about what you are. If you're still with me in this late-night descent into the architecture of your own mind, you are part of something rare. Curious minds willing to drift through the deepest questions science has ever asked. A quick subscribe helps others find this contemplative journey. Now let's continue into the space between your neurons, where thought itself is born. Chapter 4. The Synaptic Void. The space where thought is born. Here is something that should genuinely stop you. Your neurons do not touch. Every thought you have ever had, every memory, every moment of recognition, or grief, or sudden unexpected wonder, every conversation, every decision, every dream, all of it, every single cognitive event in the entire history of your inner life has depended on signals crossing a gap. Not a metaphorical gap, not a gap in our understanding, a physical void 20 to 40 nanometers wide that separates the axon of one neuron from the dendrite of the next, the synaptic cleft, and it is the most consequential empty space in the known universe. When an electrical signal, the action potential, travels down a neuron's axon and arrives at the terminal bouton, it triggers a cascade. Voltage-gated calcium channels in the membrane open. Calcium ions flood in. This triggers synaptic vesicles, small membrane-enclosed packages containing neurotransmitter molecules, to fuse with the cell membrane and release their contents into the cleft. The neurotransmitters, freed into that 20 to 40 nanometer space, diffuse across it under the random influence of Brownian motion, the same thermal jostling that moves any small molecule in a liquid, and bind to receptor proteins on the receiving neuron's dendrite. If enough receptors are activated, the receiving neuron generates its own action potential, and the signal continues. This is, on the surface, a beautifully engineered relay. Electrical becomes chemical, crosses the void, becomes electrical again. Elegant, efficient, and at the synaptic level, happening roughly 100 trillion times per second across the brain. But the elegance conceals something that should astonish us. The process is not deterministic. A given synaptic terminal when an action potential arrives does not reliably release neurotransmitters. Release is probabilistic. The probability of release at any individual synapse, depending on its type and its current state of facilitation or depression, ranges from as low as 10% to as high as 90%. There is no guarantee, there is only a chance. The brain's fundamental unit of communication is, at the synaptic scale, a gamble. This was documented in detail by researchers including Charles Stevens at the Salk Institute, who spent decades characterizing what he and others called synaptic noise, the irreducible variability in synaptic transmission that makes the behavior of individual synapses fundamentally unpredictable, even with complete knowledge of their prior history. And here is the part that took years to appreciate. This unreliability is not an engineering flaw that evolution failed to correct. It appears to be a deliberately maintained feature of synaptic design. The theoretical neuroscientist Wolfgang Mayas at Graz University of Technology has worked extensively on what he calls liquid-state machines, computational models that exploit the kind of dynamic, probabilistic variability found in real neural circuits. His models suggest that synaptic noise actually expands the computational power of neural networks rather than limiting it, enabling a form of computation that rigid, deterministic circuits cannot perform. The brain, it seems, uses its own randomness as a resource. Unpredictability is not a bug. It is part of how thinking works. Now hold this alongside what we explored in the physics of luck. In that episode, we spent time with the question of whether quantum probability, the irreducibly random behavior of quantum mechanical systems, percolates upward through the scales of physical reality, leaving its signature in macroscopic events we ordinarily attribute to chance. We asked whether luck at some deep level has quantum roots. The synaptic cleft is exactly the kind of scale and mechanism where that question stops being philosophical and becomes experimentally concrete. In that 20 to 40 nanometer gap, neurotransmitter molecules are subject to thermal fluctuations that approach quantum scales. The binding of a single neurotransmitter molecule to a single receptor protein involves electron transfer events that quantum chemistry governs. Some computational neuroscientists have proposed that the probabilistic release of neurotransmitters may be sensitive to quantum-level variability in a way that makes the classical distinction between thermal noise and quantum uncertainty difficult or impossible to draw cleanly at these scales. This does not mean your decisions are random. At the systems level, the level of circuits and networks and the integrated output of billions of synaptic events, the brain's behavior is recognizably structured, consistent, and in most circumstances predictable enough to constitute a stable personality. But it does mean that the machinery underlying those decisions incorporates, at its physical foundation, a form of genuine indeterminacy. The synaptic void is not a passive passage. It is a probabilistic zone, a threshold where the clean lines of mechanical causation blur. There is a second dimension to the synaptic cleft that becomes important as you lie here in the dark. The architecture of your synapses is not fixed. It is continuously being remodeled, a process called synaptic plasticity. And the most profound remodeling occurs not during the active wakefulness of the day, but during sleep. During the waking hours, synapses are strengthened by use. Every thought, every sensation, every learned association leaves a trace in the molecular structure of the synapse. Receptors are added to the postsynaptic membrane. Vesicle pools are enlarged. The geometry of the cleft itself changes. This is the cellular basis of learning and memory. This is Hebb's rule, formulated by the Canadian psychologist Donald Hebb in 1949 and confirmed at the molecular level decades later. Neurons that fire together, wire together. The synapse is the writing surface on which experience inscribes itself. But writing constantly without erasing produces noise, not signal. If every synapse is continuously strengthened by every experience, the brain becomes a system in which everything is equally emphasized, and therefore nothing can be distinguished. The signal drowns in its own amplification. Sleep is the erasure. The neuroscientist Giulio Tononi, whose work we will return to shortly, proposed what he calls the synaptic homeostasis hypothesis. During sleep, and specifically during slow-wave sleep, synaptic strength is globally downscaled. The strongest connections, the ones encoding the most important experiences of the day, are retained. The weaker incidental connections are pruned back. The brain wakes each morning with a refreshed dynamic range, capable once again of distinguishing signal from noise, important from unimportant, remembered from forgotten. This process is happening right now as wakefulness recedes. The synapses encoding today's experiences, the important ones, the emotionally resonant ones, the ones connected to existing networks of meaning, are being stabilized. The rest are being gently released. The void between your neurons is not empty tonight. It is being tended. It is being curated. Tomorrow's mind is being assembled right now in the space between thoughts, in the 20 to 40 nanometer gap that connects everything you are to everything you will be. Rest in that space. It is doing exactly what it needs to do. Chapter 5. The Observer in the Dark. Who is watching? We have gone far enough now to ask the question that has been waiting, quietly, beneath all of this. If the brain is electrochemical signals in neurons, if those neurons are wrapped in glial networks that process information below the threshold of consciousness, if the fundamental unit of neural communication is probabilistic, not deterministic, if the microtubules inside those neurons may be operating as quantum systems at the boundary between possibility and fact, then who exactly is experiencing all of this? Who is watching the movie of your life? This is the hard problem of consciousness, and it belongs primarily to the philosopher David Chalmers, who articulated it with unusual clarity in a landmark 1995 paper that shifted the entire field. The hard problem is not about explaining what the brain does. Science makes continuous measurable progress on that. The hard problem is about explaining why the brain's doing anything feels like anything at all. Let's be precise. Neuroscience has made extraordinary progress on what Chalmers calls the easy problems of consciousness. And again, easy in the technical sense, not in the common one. Easy means tractable, amenable in principle to scientific investigation and eventual resolution. How does the brain direct attention? How does it integrate information from different sensory modalities into a unified experience? How does it produce the ability to verbally report on inner states? How does it maintain the sense of a continuous self across time? These are difficult empirical questions, and neuroscience has sophisticated, partial and improving answers to all of them. The hard problem is different in kind, not in degree. It asks, why does any of this processing produce subjective experience at all? Why isn't the entire system just computation, efficient, complex, adaptive, entirely dark, with no inner light of experience? Why, when photons activate photoreceptors in your retina and trigger a chain of neural events in the visual cortex, do you see red? Not process the wavelength signature of red. See it, feel it, have the experience of it. No one has answered this, not approximately, not with caveats. The hard problem remains completely open. Francis Crick, the co-discoverer of the DNA double helix, who spent the last 30 years of his extraordinary career working on the neuroscience of consciousness, proposed with his colleague Christoph Koch that consciousness is associated with specific patterns of neural oscillation, particularly in the 40 Hz gamma frequency band, and that the neural correlate of any conscious experience involves a specific coalition of neurons firing in this synchronized gamma rhythm. This remains one of the most productive research programs in consciousness science. It has generated genuine insights, but correlates are not explanations. Knowing which brain activity accompanies a conscious experience does not explain why that activity produces experience, rather than simply occurring in the dark. Crick and Koch's framework is a map of where consciousness happens. It is not an account of why it happens at all. Giulio Tononi at the University of Wisconsin has developed what is arguably the most mathematically sophisticated theory of consciousness currently available, Integrated Information Theory, or IIT. Tononi proposes that consciousness is not a special property that biological brains happen to have evolved. It is a fundamental property of physical systems, identical to the quantity of integrated information a system contains. He calls this quantity phi. Any system with high phi has rich conscious experience. Any system with low phi has diminished or absent experience. The theory has considerable explanatory elegance. It accounts for why the cerebellum, which contains more than half the brain's neurons but is organized in a modular, parallel fashion with low integration, contributes little to conscious experience, while the much smaller but highly interconnected thalamocortical system appears to be where experience lives. It makes precise, testable predictions. It is serious science. It also implies, and Tononi accepts this implication without flinching, that even very simple physical systems with some degree of integrated information have some vanishingly small degree of experience. Consciousness, on IIT, is not something that appeared with neurons. It is something that becomes richer and more complex as physical systems become more integrated. It may have existed in some form in the universe before biology. Others have proposed global workspace theory, the idea of consciousness as a kind of cognitive broadcasting system, where selected information is made globally available across the brain's distributed networks, and this global availability is what constitutes the conscious experience of that information. Bernard Bars at the Neurosciences Institute developed this framework, and Stanislas Dehaene at the Collège de France has developed the experimental program around it with extraordinary rigour. The ignition of global neural workspace activity, the moment when localized processing suddenly propagates across the brain in a wave of synchronized activity, can be identified in brain imaging data, and it correlates with the subjective sense of becoming aware of something. Again, a correlation, a description of when consciousness occurs, not an explanation of why it occurs. Here is what I keep returning to. We have, in the span of about 60 years, gone from almost no scientific engagement with the problem of consciousness to a situation where some of the most rigorous scientists and philosophers in the world are working on it with genuine urgency. The number of serious theories has grown. The quality of the experiments has improved enormously. The neural correlates are increasingly well characterized, and the hard problem is, if anything, clearer and more intractable than it was when Chalmers named it. This is not a failure of science. It may be a sign that science is approaching a boundary, not a permanent one, but one that requires conceptual tools we do not yet have. The history of physics is a history of encountering phenomena that resisted every existing framework until a new framework was invented: relativity, quantum mechanics, the quantum field theory that unified them. Consciousness may require a similar conceptual revolution. But here is the image I return to, lying in the dark thinking about any of this. Right now, you are the result of trillions of quantum events unfolding every second. Electron transfers, ion tunneling, molecular fluctuations at scales where classical physics simply does not apply. Each of those events is, at the quantum level, genuinely and irreducibly random. Not pseudo-random, not random because we lack sufficient information, but random in the way that only quantum mechanics is random, in a way that is written into the fabric of physical law. And yet out of all of that irreducible randomness, there is something here that is continuous, something that remembers being you yesterday, something that anticipates being you tomorrow, something that is right now aware of the sound of this voice and the weight of its own body against the surface beneath it. That continuity, that extraordinary persistence of a single perspective across the quantum noise of the body, may be the deepest mystery in all of science. Who is watching? We do not know, but whatever it is, it has been watching from inside a quantum system of almost inconceivable complexity, navigating between determinism and chance, between ancient reptilian brainstem and quantum microtubule, between the firing of 100 billion neurons and the silence of the synaptic void. Perhaps the watching is what the universe does when it organizes itself to sufficient complexity. Perhaps experience is how matter becomes aware of the laws that govern it. You don't need to resolve that tonight. No one can. Let the question rest beside you, soft and open, the way all the best questions do. Chapter 6. The Pineal Gland. What science hasn't caught up to yet. I want to be direct about something. In mainstream neuroscience and in most popular science writing that deals with it, the pineal gland is routinely dismissed. The standard modern account goes roughly like this. It is a small, pea-sized gland sitting in the geometric center of the brain. Its function is to secrete the hormone melatonin in response to the absence of light, and this melatonin signal helps regulate the sleep-wake cycle. Important in a limited and mundane way. The mystical associations, Descartes called it the seat of the soul, multiple spiritual traditions named it the third eye, Tibetan medicine regarded it as a seat of higher perception. These are presented as charming historical errors, prescientific projections that modern biology has rendered definitively obsolete. I do not accept this dismissal. I want to explain why, carefully and scientifically. The first argument is evolutionary, and it is, in my view, the most powerful one. The pineal gland in lower vertebrates, lampreys, frogs, various species of lizard, certain deep-sea fish, is not a metaphorical third eye. It is a functional, light-sensitive photoreceptive organ. In these animals, it sits just beneath a thin region of the skull, specifically reduced in thickness to allow light transmission. It contains genuine photoreceptor cells. It directly responds to the intensity and spectral quality of ambient light. It is, in every meaningful sense, an eye oriented upward toward the sky. In the course of vertebrate evolution, as skulls thickened and cranial complexity increased, this organ migrated inward. In mammals, it lost its direct photosensitivity and became what we now call the pineal gland, internalized, connected to the visual system only indirectly, through a multisynaptic pathway involving the retina, the suprachiasmatic nucleus, and the superior cervical ganglion. But consider the evolutionary logic here. Structures that serve no function are eliminated. The metabolic cost of maintaining a specialized gland in brain tissue, supplying it with blood, maintaining its cellular machinery, protecting it within the skull, is not trivial. Evolution has had tens of millions of years to eliminate the pineal if it served no purpose beyond what any diffuse tissue could accomplish. The pineal gland is present, precisely located, and structurally distinct. The null hypothesis, that it is a vestigial irrelevance, requires the assumption that evolution simply failed to clean it up for tens of millions of years across the entire mammalian lineage. That is not a satisfying scientific position. We should at minimum keep looking. The second argument is biochemical. The pineal gland's production of melatonin is far more sophisticated than the word hormone secretion suggests. The pineal maintains an exquisitely precise internal clock, operating in coordination with the suprachiasmatic nucleus, the brain's master circadian pacemaker, and responsive to environmental light cycles via its indirect retinal connection. Its output does not merely make you sleepy. Melatonin is involved in immune modulation, in the regulation of reproductive cycles, in antioxidant activity, and in a range of cellular repair processes that depend on the timing of its release with a precision of minutes, not hours. The regulatory role this gland plays across the body's physiology is sophisticated enough to suggest a structure of genuine biological importance, not a vestigial footnote. The third argument is structural. The pineal gland contains cells called pinealocytes, and when their ultrastructure is examined closely, and when their molecular biology is characterized, what you find is striking. Pinealocytes contain proteins that are, in biochemical terms, almost identical to proteins found in the rod and cone photoreceptor cells of the retina: S-antigen, arrestin, transducin, and recoverin. These are proteins whose known function is phototransduction, the molecular process of converting light into neural signals. The pineal gland in an adult human brain does not sit where light can reach it, and yet it carries the molecular machinery of vision. David Klein at the National Institutes of Health, who has devoted decades to pineal biology, describes it as a transformed retina, a structure whose photoreceptive heritage is so deeply encoded in its molecular architecture that it persists even after the direct photoreceptive function has been extensively modified. Klein does not make mystical claims. He is a careful, rigorous scientist, and his characterization of the pineal as a transformed visual organ is, within the mainstream literature, an accepted description. What this tells us is that the pineal gland is not simply a melatonin dispenser that evolution forgot to dismantle. It is a structure with deep evolutionary roots in photoreception, an extraordinarily precise regulatory role in whole-body physiology, and a molecular biology that we are still, in genuine honesty, working to understand. In traditional Chinese medicine, and specifically in the Heidelberg model, developed by Dr. Henry Johannes Greeten, which grounds ancient clinical observations in modern physiological language, the structures that classical practitioners identified as centers of regulatory intelligence do not map arbitrarily onto anatomy. They correspond, often with striking precision, to sites of genuine physiological importance that Western medicine arrived at through entirely independent investigation centuries later. The convergences are too consistent to dismiss. I am not saying the pineal gland is the seat of the soul. I am not saying it produces mystical experiences or connects us to transcendent realities. I am saying we have a gland sitting at the geometric center of the human brain, evolutionarily descended from a functioning eye, carrying the molecular signatures of photoreception, performing regulatory functions of substantial complexity, whose full biological role we genuinely do not yet understand. And the scientifically honest position is not dismissal. The scientifically honest position is we should keep looking. The history of medicine is full of structures that were dismissed before the conceptual tools existed to understand them. Glial cells were the brain's glue for a century. The enteric nervous system, the vast neural network of the gut, was considered a simple relay until it proved capable of operating independently of the brain. The fascia was connective tissue filler until it became clear that it contains mechanoreceptors and plays a role in sensory processing that physical medicine is only beginning to map. The pineal gland may be on that list, and if it is, the ancient physicians who placed their hands on the body and felt something real happening at its center without electron microscopes, without fMRI, without the entire apparatus of molecular biology, may turn out to have been, in their way, observing something genuine. As you drift deeper tonight, let this rest softly at the edge of awareness, at the center of your brain, older than almost everything else in it. A structure sits that was once oriented toward the sun. Whatever it is doing in the dark, whatever it has always been doing, it has been doing it for longer than the human species has existed to ask why. Returning to the surface, falling gently back into the dark. We've travelled far tonight, from the ancient brainstem, keeping you alive with an authority and competence that predates consciousness itself, through the bioluminescent forest of the glial networks, doing their extraordinary work below the floor of awareness, into the quantum geometry of the microtubules, where serious science flirts with the most fundamental questions about what experience is made of, across the probabilistic void of the synaptic cleft, where thought assembles itself from the raw material of chance, into the unanswerable question of the observer, who is watching and why watching feels like anything at all. And finally to the quiet, underestimated mystery at the geometric centre of your skull, doing something in the dark that we have not yet found the language to fully describe. What I hope you carry from this journey, not as an idea to analyse but as something felt, something settled into the body, is this. The strangeness is not elsewhere. It is not in distant galaxies, or in the mathematics of string theory, or in the particle accelerators buried beneath the Alps. It is here. It is the tissue you inhabit. It is the biology that processed every word you heard tonight and converted it into whatever experience you are having right now, in this moment, in this body, in this dark. You are stranger than you know, more ancient than you remember, more quantum than you feel, and the most profound mystery in all of science, the question of why any of this is experienced by anyone at all, is not a question about the universe out there. It is a question about you specifically right now, and tonight, as you surrender to sleep. As the thalamus dims its gate, and the reticular activating system quiets, and the glymphatic tide begins its faithful nightly work, you are not disappearing. You are dissolving into the deeper architecture, the one that does not need your permission to function, your awareness to operate, or your understanding to continue its extraordinary ancient quantum work. You are safe in the deep architecture. Breathe softly now. Let the ancient brain carry you. Let the quantum dark receive you. Let the forest do its work. This is sleep entangled, where the impossible physics of the universe and the quiet wonder of the conscious mind drift together gently into the contemplative dark. Good night.